Okay, great. So, whoops, sorry. Let's go to Isabell, who's a senior PhD student in the Ebersol group, and during her first couple of years here, she worked a lot on this, also with George, these FDR considerations, and now we're going to talk at how these are going to be propagated when you start going to protein level or large experiments, and also a little bit about feature alignment between runs.
Which one is this?
Is it 5?
Okay, perfect. Okay, good morning, everyone. So now I will basically extend on what Ludovic was just presenting in the last hour. So I will extend a little bit on FDR control in large DIA data sets, and then talk a little bit about feature alignment across large data sets to increase the completeness of your data set and the to basically get consistent detections across many samples. So first of all, I'd like to put this peptide-centric scoring and FDR calculation a little bit into context. So to quickly recap what Ludovic talked about is, first of all, he was mentioning how we perform automated chromatogram extraction and peak group scoring for each of the targets and the decoys in our spectral library. Then we perform the semi-supervised learning to actually calculate discriminant scores that can separate targets from decoys. And then the statistical scoring is done using a story-Tipshirani approach to get Q values for all of your targets and decoys in the spectral library. Yes. So some things that might come into your mind are, okay, so what we're doing so far, or what we've been talking about, is to perform the error estimation on the peak group level or peptide query level. And the other thing is we are talking about targeted extraction and scoring of one single sample. But if you think about the data sets that you are analyzing in your lab, you probably are interested not in peak groups, but you want to report peptides and proteins. And also, most of the times you don't analyze one sample, but you actually are interested in a large data set, including multiple samples. So now the question is if this makes a difference. And the answer is yes, and it depends on your experimental setup and the way that you analyze your data. So three important considerations are, first of all, the size and the comprehensiveness of the spectral library. Second important consideration is the number and the heterogeneity of the data set that you study. And last but not least, there is an important relationship between the proteins present in your sample and the proteins that are represented in your spectral library. So the way that we started off with analyzing DIA data in the peptide-centric or targeted approach was coming from SRM. So here we generate a very sample-specific spectral library, which means that we have a fairly homogeneous set of samples, for example, coming from the same cell line. And then we analyze them with GDA and get a sample-specific spectral library, the way that you generated it in the tutorial yesterday. And then we perform a targeted DIA extraction. But now, sometimes you don't have a very homogeneous data sample set, or you just get to more complex data sets. And then you end up in a scenario where you have a more complex spectral library. For example, when you are working with heterogeneous samples, so you have already a larger and diverse, I would say, study-specific spectral library. And the analysis that you perform is, in this case, already a little bit less targeted compared to our standard approach. And then there is another scenario that you can actually want to use a repository scale library, like I think Ben mentioned a few yesterday. And in this case, you have a very extensive spectral library, but you might be analyzing only a small sample set that is only representing a very small fraction of what is actually represented in the spectral library. And the important difference between these two cases is that the discriminant score distributions that Ludovic has been talking about look really, really different between these two cases. So for sample-specific libraries, we usually have a discriminant score distribution that looks something similar to this. So here in gray are the decoys that distribute around 0, and we have targets where most of the targets are detectable and score very highly, and we have only a very few, like large, small proportion of targets that fall into the decoy distribution. On the other side, if you have a very comprehensive spectral library, then what you can frequently see is that the proportion of true targets is very small, and you have a very large fraction of false targets. And this is what we call pi 0, or the fraction of false or non-detectable targets. So this can be estimated by the number of non-detectable targets over all targets. And the very important point here is that if you have the larger your pi 0 is, the higher is the statistical burden, meaning this, the correction that you can do that Ludovic was talking about is very tiny. So this has several effects. So first of all, when you move to large pi zeros, the peptide query FDR and the protein level FDR start to diverge. So if you control the FDR in your data set at 1%, you will see that you might end up at 5% protein level FDR. So why is this the case? Here's a simple toy example. We have detected several peptides in our data set, and we allowed 15% FDR. So in this toy example, we have 3 false positive peptide detections. Now, if we move to the protein level and aggregate our peptide identifications, we will see that true peptides tend to cluster together and they match to the same protein, while the false detections are by definition random, so you might end up with like a lot of false positive proteins for your peptides. So when you then look at the FDR on the protein level, you actually end up at 42% FDR in this toy example. So therefore, it is very critical that you control the FDR not only on this peptide query level or peptide level, but that you also report protein level FDR if you at the end want to work with proteins, which most of you probably want to do. And one way you can do it is by basically trying to get an estimate on the protein level by, in our case, we have implemented a version where we take the best scoring peak group per protein as a surrogate for the protein. And what you can then get is a discriminant score distribution on the protein level. And here you can already see that the pi 0 on the protein level is much smaller than on the peptide query level. So, and then what you can do is you can use this discriminant score distribution on the protein level and perform Q-value estimation by the Story-to-Priani approach, same as on the peptide query level. I just wanted to ask if you have any questions until now. Okay, I continue. So I think this is covered. The second problem that can come up is that you don't usually look at one run, but you look at a large data set. And what can happen is that if you do multiple different analyses, your error can accumulate across runs. So if you have 1% peptide query FDR in your single run, if you accumulate across runs, you might end up at like 20 runs. You will have 25% FDR in the global data set. So the toy example here is again that you have let's say a spectral library containing these 7 proteins, and each of these lines here represent one sample, and in each of these samples you controlled your FDR at 20%. So you have one false positive detection per sample. But if you at the end want to report your global set of detected proteins, if you just simply add up all of the protein detections across the runs, you will end up at 40% protein level FDR in your global result. So we have thought about this problem quite a bit in our lab, and we have come up with 3 different questions that you can answer, similar to the questions that Mike mentioned between spectrum-centric and peptide-centric. We now want to differentiate even further, depending on how many samples and what specific question you want to answer. So one question could be if a protein is detected in a very specific sample in your data set. And this is what Ludovic has been presented in the lecture before. So you're really just interested in one single sample and you want to know if a certain set of proteins are detectable. And the way that this, we call this the run-specific context. So the way that you calculate D-scores is on a per-run basis. So here is just the toy data matrix. So you have the different runs, 1, 2, 3, your analytes, and what you can then do is basically do a discriminant score distribution for run 1. And what you can see is here at a D-score of 3.4, if you do the Story-Shirani Q-value estimation, you get a fairly good Q-value. Now, if we look at the second run, at a similar D-score, but we have a different distribution of true and false targets, and you will get already a way worse Q-value. If your pi 0 is getting even bigger, you will see that a similar D-score will end up with a way worse Q-value. So here, this means that the same D-score can have very different Q-values. But if you're interested only in one sample, this is totally fine. But now the question is, okay, what is if I want to determine in which subset of samples I can detect a protein? And for this, we use this experiment-wide context where we generate an accumulative discriminant score distribution. So we take all of the identification or detections of our protein across the entire data set, and we get an accumulative discriminant score distribution. And when we then convert the D-score to the Q-values, we will see that the same D-score is also resulting in a comparable Q-value. And this might be beneficial if you have very heterogeneous data sets and you want to assure that when you have like the same peak, like visual peak quality, that you also get a similar Q-value. But now the question about error accumulation was coming up when aggregating the results across a large data set. So the question we ask is if a protein can be detected at least once in our study. And for this, we have this global context. And what this means is that we can basically take the best protein detection across the entire data set as a surrogate for the global detection. And then we will get an optimal best D-score distribution. And then you only get one Q-value for each analyte. And by using this, you can generate a master list of detectable proteins in your study. And the way that we are using this in our lab now is that we run this global context model to generate this master list and then filter our data set that we either analyze with a run-specific or in the experiment-wide context. A practical implementation where we have included this protein level and across-run FDR estimation is PyProfit, or like an extension which we internally call Jumbo PyProfit, which means that we, is basically a Python reimplementation of the mProfit algorithm that Ludovic has presented, but it is has a few additions. So First of all, the semi-supervised learning is conducted not only on one run, but it's actually done across all of the runs together so that you use the same score weights across the entire data set. And then we perform the FDR estimation on peptide query, peptide and protein level, and you can choose the different contexts depending on the question that you ask. And this code is, or the software is available on the GitHub repository of PyProfit, and it's documented on the Open SWATH website, and we will also use this in the tutorial 4 on Thursday. So now I want to show you a case study where we used this model and where it was very important. This is an example of the SWATH-MS Interlaboratory study, and the experimental design is not entirely important, but what is important is that this data set includes 229 DIA measurements that have been measured in different sites around the world, but they have all been centrally analyzed with OpenSWATH and PyProfit. And the questions that we wanted to ask are, what is the impact of using spectral libraries with varying size and sample representation, and what are the implications of different error rate control strategies on this data set? And what's important is that this is like 229 times the exact same sample. So the three libraries that we used to analyze the data were, first of all, the combined human assay library that I think Ben mentioned yesterday in the library generation talk, which was generated based on 331 different DDA injections from several different human sample types, including HEC293 cells. Then we used a HEC293 subset of the combined human assay library, which was only then generated from 134 samples. And last but not least, we used the sample-specific library, which was only generated on 6 injections of these HEC293 cell lysates. And here you can see that the combined human assay library contains roughly 10,000 proteins. The HEC subset has a set of roughly 6,000 proteins, and our sample-specific library only contains 2,500 proteins. And if you look at the discriminant score distribution in the global context of this Interlab study, HEC293 cell lines, you can see that the combined human assay library results in a very large pi0. Here, this is depicted in this yellow line. So you see that we have a large proportion of false targets. The HEC293 subset is matching our samples way better. So the fraction of false targets goes down to 0.3. And for the sample-specific library, we have almost no false targets. So here in this dark red, you can see this is almost 0, and all of the targets are detectable. And now the question is, how does the FDR strategy impact on the analysis? So if we use this very large combined human assay library and we control the error only on the peptide query level, you can see that here on the x-axis is the number of runs, and on the y-axis is the cumulative number of protein detections in the data set. And you can see that if you only do this peptide query level FDR, you accumulate detections until you almost reach the full library size. If we perform protein level FDR estimation and control at 1% protein level FDR, the accumulation of errors is already going down quite a bit. And only if we do a global protein FDR control, you see that we actually see what we want to see, which is that we don't accumulate any detections across the runs because they should be all identical. And if we now compare this to the other, yeah.
Were you ever comparing with a peptide, global peptide FDR?
So
Unique peptide level FDR?
Yeah, so we have calculated it. I don't know if we have actually looked at the figures. I would also assume that this has a similar effect. So I think it's essential that you do protein level error rate control, but maybe it's not essential that you do it on in the global way. But I think what is important is that you at some level do a protein level FDR control. So you can do this in the experiment-wide context, which we definitely did. But you can, I don't know, for me it makes sense to also do it in the global context.
Yeah, no, I think the more important one is the global context, and whether it's protein or peptide is less important. But we don't have the line, so I don't think it's.
Yeah.
So I think that would depend on the library size, right? In a very large library, that would make a difference. In a small, like sample-specific library, I think it would make less of a difference.
Yeah.
Because you risk even a small number of kind of like false peptides in the large library, because we're taking each protein as represented by the single best peptide, these kind of accumulate. So I think it would depend on the library.
I also think that we would still have the same problem as before. Okay, but now, as Ben said, this really much depends on the library. So here, this was now on the combined human assay library, which is fairly large. If we look at using only the HEC293 subset of this very large library, then you can see that the error accumulation is not as bad as for the full library. And if you use a sample-specific library, as with the dark red line here, you can see that it essentially doesn't really matter what kind of error rate control you perform, because all of your targets are represented. You cannot accumulate false targets because they're not even represented in your library. And here is just to give you an idea of what kind of, like, how many proteins and what is the overlap of the proteins that we detected with the different libraries. And you can see for the combined human assay library and the HEC293 subset of the combined human assay library, we detect a similar number of, like a little bit, no, it's, what is it, almost 5,000 proteins. And you can see that there is a very small difference. And this can be explained by, here for the combined human assay library, you have some more opportunities because you have a larger library, and a few of the proteins that are not represented in the HEC subset might actually be really there in your sample. And these proteins are the ones that you don't even have in the HEC library. On the other hand, in the HEC library, you see a few proteins that have not been detected with the combined human assay library, which is basically because you get a little bit deeper because you don't have to correct as much for the multiple testing. And what you can also see for the sample-specific library, we get way, way less protein detections, which means that we see what we have in the library, but we don't fully explore the entire, like, everything that is there in the data set. So another example, which is probably a little bit more striking, is an application to the human blood plasma cohort, including 246 blood plasma samples of 116 individuals. And we analyzed this data set with both the combined human assay library and the plasma-specific library. And what you can see is, as you would expect for human plasma, you only see a very, very small set of the proteins that are represented in the combined human assay library. So your target distribution here is almost completely overlapping with the decoys, and you have very, very, very few true positive targets. And for the plasma library, you can see that you have a much better recovery of the proteins represented in the library. However, if you do global protein FDR control and analyze this in a good way, you actually see that re-extract a similar set of proteins. However, if you don't control the error rate appropriately, if you use the combined human assay library and filter again only on peptide query level, you can accumulate detections until you reach a detection rate of 3,500 proteins in human plasma, which is, I hope you agree, not very plausible. So what are the things that we learned from the study? The two questions that I asked at the beginning to the study was, what is the impact of the spectral library on with varying size and sample representation? And we figured out that there is definitely a trade-off between the comprehensiveness and the multiple testing burden, which means that when we have a large library, we have a gain in opportunity, because we have a larger set that we can actually extract and find. However, smaller sample-specific libraries benefit from having like less statistical burden. The second question was, what are the implications of the error rate control strategies? And we realized that for sample-specific libraries, error rate control on the peptide query level, as we have done in the past, is sufficient. But as soon as you go to large cohorts and using comprehensive spectral libraries, it is essential that you perform protein level FDR control and that you assess the error rate across your entire data set in a global fashion. So now the question is, of course, what library should you use for your study? So I think in general you could say that for the sample-specific library is very useful for studies where you have a well-defined set of samples which are very similar to each other, so for example coming from the same cell line. However, if you have a heterogeneous sample cohort, you will immediately get to a stage where you have or need to use a comprehensive library, and the representation of your samples in the library will be not as good. Then you get to this side of the scope. And second application is maybe you don't want to go through the hassle of generating a DIA library for your DIA analysis, and you want to use a library resource, and in this case you are also rather on this side. However, what is very important to keep in mind is that it doesn't make a lot of sense to use a library that very poorly represents the sample that you analyze. So if you, for example, want to analyze the urinary proteome and use the database that or spectral library repository that doesn't even contain any urinary samples, you will be very badly off. So there is definitely a trade-off between the comprehensiveness and the statistical burden, and I think it's important that you keep this in mind when choosing the way to analyze your data. So with this, I'd like to get to the second part of the presentation, which will be about, yes?
Could you then maybe comment on fractionation of your samples for the spectral library? Because we ran into it and then had this impression that I have a very high pi zero fraction basically, and not so great in analyses.
Okay, so generally, from my experience, fractionating the sample is generally beneficial. So if you
So really deep fractionation, so we use them really like 20 fractions, and I had the impression we couldn't really find them then in a regular DIA run.
I think this depends on the DIA acquisition that you use, and I don't know, maybe someone else has, Ben, do you have some comments on this?
I don't think you expect to find everything you find in the deep fractionation library in the single-shot DIA analysis, right? Yeah. So, but I mean, I would say that's not even the goal. I mean, what you're trying to do is, so what I was trying to say yesterday is basically, if you do a single-shot DDA analysis of the sample, you probably don't identify everything that you can get out of the DIA data. So what you do is then do a fractionation, and what's probably happening is you're going to mostly overshoot, so you'll have a larger set of peptides than actually identified in the fractionated sample that then actually exist in the DIA sample. So that means most or a large part of this deep fractionated library is not going to be detectable in the DIA sample. So that's not a big surprise. But it still might be a reasonable thing to do in order to kind of exploit as fully as possible what's in the DIA data. But if you do that and you have now this large library where you're looking for lots of things that are not going to have a signal, you're never going to have to be able to find a signal for, then you're into the domain where you need to make these type of precautions that Isabell is talking about.
Okay. Any other questions? Okay. Okay, so now I will talk about the software called TRIC, which stands for Transfer of Identification Confidence. And the goal with this processing step is to correct for nonlinear chromatographic distortions between different runs in a large data set, and to minimize the effect of outlier runs to get a fairly comprehensive and complete data matrix. And this the goal of this tool was to actually really be able to scale up the analysis to very large data sets. So the way that TRIC works is by, first of all, generating a reference-free alignment, which means that you across all of your, between all of the runs in your data set, you perform, you calculate the pairwise distance matrix by performing a linear regression between each two runs. And then you can use the distance matrix to construct a minimum spanning tree, which will contain all of your samples as nodes, and the edges between in the tree are representing the retention time difference that you calculated here. Once you have generated this tree, which represents how similar your different runs are in terms of the chromatography, you can do a nonlinear retention time alignment, which means that you basically, for adjacent runs, adjacent samples in your minimal spanning tree, you can do a nonlinear regression, for example, by spline or Loess, to get a good alignment between neighboring runs in this tree. And what you can then do is you can walk through your minimum spanning tree by starting so basically you do this for each peptide query peak group. So when you have like a peptide query in your library, you start with the best peak group that was detected in your data set, and you walk through your spanning tree and find the according peak groups in the neighboring runs. And what you can then do is you can transfer the identification confidence. And now the question is, what does this mean? This means, first of all, that you can use the information of all the runs in your study to increase the confidence of detections that are probably not so good in a single run. So if you, in this example here, have 4 different runs, and in all of the 4 runs you have detected a peak group that has different Q values. So here you have a very good Q value, you have another good Q value, here you don't have a very good Q value. So this peak group will not, in the standard workflow, pass the 1% FDR threshold. But as soon as you align it with these other runs, TRIC says, okay, maybe we can be more confident about this peak group because we know that in all of the other runs, or in many of the other runs, we have identified this peak group very significantly. So you can say, okay, as long as we have a very good detection of this peak group, we maybe allow a 5% Q value in other runs, which have a very good retention time alignment. The second thing that you can do is you can increase the consistency of the peak selection between your runs. So I think Ludovic already showed an example where you can have multiple different peak groups in your retention time window. So here you have 2 peak groups that look fairly good, and if you look at this one run specifically, you will probably go for this peak group because it has a better, so at the end you would use the Q value of these different peak groups, right? But here you would maybe choose this peak group. However, if you do the TRIC alignment, you will see that the alignment actually predicts the peak to be in this region. And then you can basically tell the algorithm to choose this peak group instead of the other peak group, because it's more consistent with your other runs, and this way you have a better consistent detection of the same and quantification of the peptide from the actual same peak group. And last but not least, you can revisit missing values in your data matrix. Basically, using this retention time alignment, you can find areas where there should be a peak based on the other runs. And you can integrate the noise level in this area to actually get also value for the missing runs. However, this should be treated with caution, because it might be that you have some interfering signals in the same region, and you should always, if you want to use this requantification, to check manually that you don't get like way higher signals than all of your actually detected peak groups, which is kind of important. And this is just to show that how TRIC actually really improves your data. So this is a validation data set where 7,000 peak groups have been manually annotated. And you can see that if you use the naive open SWATH workflow, you would get a recall rate shown with a black line. And if you use the TRIC alignment, you get a much better recall, and also the error rate estimation gets more precise. So what you can see here in the naive, so all of these 4 different modes were reported at 1% FDR, but the naive analysis, the actual FDR in this manually annotated data set was almost 2%, while when you do the TRIC alignment, you go down with the FDR quite a bit, because you also remove peak groups that didn't fall within your retention time alignment. So these peak groups can also be kicked out in this. And what you can also see is that here you have the retention time on the x-axis and the retention time difference to the manual annotation on the y-axis. And this is before TRIC normalization, and this is after the TRIC normalization. So this is really improving your signal detection. Here, this is also just like the cumulative number of peaks that could be assessed at the specific retention time difference range. So after the linear retention time alignment is already improving the peak detection, and the non-linear retention time alignment is further improving. So using this TRIC alignment in a larger data set of yeast cell lysates, you can see that if you do no retention time alignment, these are just the number of prototypic peptides per run, and you can see that you have very variable numbers of detections across all of the different samples, while if you use the retention time alignment, you can actually get way better way better and complete matrix of your quantifications. So with this, I'd like to summarize that I've presented the context and scope of target DIA analysis and how it matters in a way that spectral libraries always come with a trade-off between the comprehensiveness and the multiple testing burden. I hope I could convince you that it is very essential, if you use comprehensive libraries with large pi zeros, that you should perform protein level and global FDR control. And finally, DIA data consistency and completeness can be improved by using a cross-run alignment to get better matrix completeness and to transfer confidence between the different runs. Okay. Thank you for your attention. And if you have any questions, I'm happy to answer, or you can also come and talk to me in the next couple of days.
Thanks again. Lauren.
Yeah.
It doesn't do anything, it just records the audio. But it's good if you use it though.
Yeah.
Would it actually make sense to do it before you do the FDR control? To have like better retention time alignment and then do the FDR on the aligned retention times? In like kind of doing two iterative searches?
Yes. So yes and no. So for the global FDR estimations, it doesn't make a difference because TRIC also uses the best peak group per run to start the guidance tree, and therefore the global FDR will not change. So this I've tried actually. For the per run basis, there could be a difference. So TRIC is reassessing the FDR. I'm not sure if anyone tried to actually rerun PyProfit afterwards. So one thing is that TRIC requires to have some statistics already to begin with. So it selects the peak group based on the Q values. So I'm not sure how valid it would be to actually reiterate this, but might be interesting to try, yes.
I mean, if you do change which peak group you're picking in a given run, it doesn't actually change the discriminant score. Actually, the discriminant score for that other peak group would be worse. So it's not that the peak group is being rescored entirely in the scoring model. It's rather that you're just picking a new one in to be consistent with the retention time of the rest of the runs. So I don't know how easy that would be, actually. I think it actually might make it worse, right? Yeah.
Do we consider the entire RT range to select these best peak groups with maybe roughly five peak groups per 10-minute window, something like that, or you just go based on some kind of scoring pattern?
Can you repeat again?
I mean, when you select these peak groups for alignment,
Yeah.
alignment, do you consider the entire RT range?
No. So we usually select, I think, a 30 to 60 second window even only. So we take the best peak group, and then after the alignment, so first the alignment is done, and then it's a very narrow window that you use.
And I'm selecting just to choose the peak groups, like how many peak groups are selected for alignment, and whether they are across entire RT range.
So for so the peak groups that are reported initially are so in the open SWATH search, for example, you select usually a window which is a little bit wider. So this is I don't know. Is it usually like a few minutes?
5 to 10 minutes,
Yeah.
something like that.
5 to 10 minutes, and then basically what we do usually is like take the top 3 or top 5 peak groups in case there are multiple. We take the top 3 or top 5 peak groups and transfer them to TRIC, and then you can basically TRIC assesses if you have initially used if your best peak group is actually the one that is aligning, and if it's a different one, it will re-rank the peak groups within this window. Yeah.
I've got a couple of questions, so feel free to stop me and others. What's the computational complexity of TRIC in terms of number of samples or anything like that? Because I'm told we're having trouble running it with 1,000 samples or something like that.
That's a good question. So I think it was run already on 1,000 samples, but it gets computationally heavy if you want to do the requantification. So without the requantification, it's fairly scalable. With the requantification, it's a little bit more tricky.
I think it depends a bit on the alignment method, right? So you can choose between different alignment methods, and some are native to Python, but there are also some Cython parts of this, and these are much faster. So I think these Cython parts were implemented to solve this basically 1,000 run problem. So I'm pretty sure we've aligned like 1,500 samples, something like this, and that might take a few days, but I think it it's basically working. So I would look at the alignment method, and might not necessarily be the default one, might not be the fastest one.
What about 3,000 or 5,000 or?
That we haven't done yet, but it depends also, it's probably a function of the file size, right? So if these are like short gradient runs with less features inside, then it might scale a bit more. If they're long gradient runs with a lot of features inside, yeah, I think, as far as I'm aware, the biggest we've done is 1,500, right? I think, but yeah, this you'll have to try.
So you said before that you think the same D score should have the same Q value. How attached are you to that interpretation? Because from my perspective, if pi 0 changes, then that's, I mean, it's optimal to have it done on a per sample basis, and you'll lose something, I would think, by going on experiment wide.
Yes, so there are different considerations for this. So depends on what kind of data set you're looking at, right? So generally, I think you're absolutely right that it might be, so it's generally beneficial to do discriminant score calculations and Q-value estimation per run, because you will get the most out of the specific data set. And if you do it experiment-wide, you will basically penalize the very good runs, and you will basically increase the confidence for the lower content samples. There are several applications where this is beneficial. So I've, for example, worked with data sets where you have like you do fractionation and you want to look at each of the samples, and you know that you have, like, you should consistently detect, like across several consecutive fractions, you should detect the same proteins. But if you like, each of these fractions could have a very different content of proteins, and your pi 0 might vary quite a bit between the fractions. But if I have a good-looking peak group, I want to definitely keep this peak group, and I'm not so much concerned about the actual pi 0 and like keeping it as optimal as possible. But I want to have like one peak quality resulting in the same FDR. It's more intuitive if you do it like this. I think there are different pros and cons to using either method. Depends on the question that you have.
We did have some issues with like weird data sets which had a lot of varying samples, and then the Q values are supposed to be comparable, right? When you do this run specific one, conceptually they're supposed to be, you should be able to directly map them, but the question is whether they're really perfectly calibrated, and do you end up with ranking problems then? So this experiment-wide one is maybe a bit more robust to this.
Is there a possibility that PyProf is also going to be implemented in Skyline, or?
Oh, it is. So, well, the sort of simple version of it is the jumbo PyProfit. We have to ask Brendan. This is coming, right? Or when Brendan?
Would that pose a problem for trig alignment? If you, for example, want to align them, am I aligning then all my healthy samples, all my disease samples, which is one sample that's similar between? Would it be a problem?
Generally, it shouldn't be a problem. So this might get a problem if you want to do this requantification, because you might have different backgrounds. But if you didn't detect a peak group in the first place, it will not do anything, right? So you need to have at least, so usually what we do is you say, okay, you want very, very confident kind of seed peak groups, and then you align up to an FDR of 5% or 10%, but you don't go any higher. So you will not boost complete nonsense peak groups. So this way it doesn't hurt if you have very different samples and you align them. This should be fine.
As long as you have enough peptides that are common, right? So that you can do this first.
Yeah.
So why would you want to do FDR in the global context? So my background is I'm a statistician. I'm not primarily interested in identifying this protein present in the experiment, and that's
So it's a very good question, but the thing is that in a lot of studies, what people report is, oh, we have analyzed so and so many samples and we can detect 5,000, 6,000 proteins, and then people go ahead and do a differential expression analysis. And the problem is if you don't do this global FDR control, this one number that you state, which is this 5, 6, 10,000 proteins, will have a lot of false positives in there. And for this kind of statement, it is kind of important to do it. And also, if you do a further analysis, so our goal of taking this global list to filter your experiment-wide or your run-specific samples is to basically also kick out false positives in this data matrix to begin with, right? So it's not only the reporting of the number, but it's also to keep the matrix at a good shape.
Can I just follow on to that? Another thing I've seen with the global FDR. So if you have, say, one peptide that's present once in, say, 1,000 mass spec runs, but the p-value for that is 10 to the negative 6, that's
Yeah.
what we count as very significant. I don't really consider that
Yeah.
to be plausible. So is that a
Yeah.
problem with the methodology, that it takes p-values as opposed to, say, a threshold that must be present in 10% of the runs or something like that?
Yes, so this is another thing that you could do. You can use the, so this is something that you can do afterwards in the post-processing, which we sometimes do. We say, okay, if we have a large cohort, we actually only consider peptides or proteins that are detectable in at least 20, 30, 50% of the samples, depending on what your experimental design is. I think it would be definitely good to implement a model that doesn't only depend on the best possible peak group, but actually considers all of them. And there's now a tool for DDA called TRIC that was presented this year at ASMS from Lukas Schall's lab. And they are basically integrating posterior probabilities across all spectra, to peptide level, to protein level, across runs, and up to the differential expression analysis. And I think this is something that we hopefully can also integrate in DIA analysis in the future, because I think this will definitely make our statistical approaches much better.
So would you consider that kind of filtering to be a replacement for FDR in some sense?
Mm,
The filtering based on number of proportion of detections in them.
So I think heuristically it does a similar thing, right? But you lose the information about what the false discovery rate is. So you could imagine doing it before the FDR calculation, actually. So I think you could apply whatever filters you would like, as long as they're symmetric to the targets and the decoys, and you could compute the FDR after that. But doing this kind of frequency-based filtering after the FDR calculations, well, then you sort of lose the information about what your FDR is, and that seems like, so people do this heuristically because they know intuitively it makes the data look better, right? But they don't know how much better or how much worse. So.
So this is probably controversial, but my response to that would be, why do I want to have that FDR interpretation results if the end result of doing some kind of data filtering is much the same?
Well, but you don't know how much the same it is, right?
True, but the point is that that hypothesis test that this is intending to correct for is not the thing we're primarily interested in.
Well, in some cases it is, because it depends what your philosophy is and what you're going to do with the data. If you want to say, okay, I quantified, 5,000 proteins, and 500 of those are differentially expressed, and now I want to validate 100 of them. Well, if there's a 50% false discovery rate in that, then you're spending a lot of time on nonsense, basically. So I think it depends what you're going to do with the data afterwards. Yeah.
Surely wouldn't you assume that if something is a false positive, then it's most likely going to be weeded out during the DE analysis, where you're not going to expect that if something's inconsistent or variable across the samples, a false positive isn't necessarily going to be consistent.
I mean, that's why Isa brought up this joint probability model, which I think is a very good idea. But it's also possible for you to assign the wrong identity to something and it to be differentially expressed, right? So now you end up with something that you say is different across samples, but it's not the thing that you think it is.